Hello everyone. I don't see anyone on the stream yet, so I'm going to wait for you guys to say hi, okay? We'll start in 5-10 minutes after most of you sign in, okay? Looks like only 10 people have signed in, so I'm not sure others are there or not. So let's wait, okay? Okay, as people are joining, I will just get the recap of where we left on Monday, and we'll continue to talk about metagenome workflow. So, where we left was we know what metagenome is. It is just a cocktail of many organisms, DNA from the cocktail of many organisms that combined gives you a metagenome. So you could, as long as you can extract DNA out of a sample, either out of a spoonful of soil or from gut microbiome or from air or from water, as long as you can, um, as long as we can get going, you're able to hear, right? So as long as we can get the DNA extracted, sequencing machines don't care. So as long as you can extract the DNA, prepare the library, and put it for sequencing, it will sequence it. So the whole thing then comes down to bioinformatics. What do we do in the computers to now say which organisms did get sequenced? How do we separate them? How do we assemble them? And how do we characterize them? That is part of metagenome. So, in a way, you can think of metagenome as a regular genome, single organism genomes as a subset of metagenome. Because in metagenome, you could have a eukaryotic sample as well. For example, when you're sequencing plant root, you get plant genome as well. Along with it, all the microbes comes in. So that's also part of metagenome. So, in any event, so to characterize the organism within a DNA sample that you sequenced is metagenome. Single organism sequencing and assembly of those things becomes a subset of metagenome project. That's why we started with metagenome, so we can get a big picture of what's going on under genomics. And in a way, metagenome is also interesting because, as we discussed, microbiome really rule the world, and we have 39 trillion microbes on our body. Every day we are carrying them around. They are keeping our skin shiny, and they help us digest our food. So it also causes disease sometimes. So metagenome is a big part of human diseases right now. So I'm going to switch now to, and also what else we did was we know how we can sequence really small fragments, 300 base pair fragment, and sequence a large number of them, so we can assemble them and come to know what is there in the metagenome that we sequenced, which organisms were there, how do we characterize them. So I'm going to switch to the PDF mode right now, PowerPoint mode. And so the metagenome workflow. Okay, this is the metagenome workflow we are going to follow, and each step of it will become a part of a stream in this lecture. So what happens is you got the sequenced reads. I told you yesterday that reads are coming. Each reads, many fragments get picked up, and each reads come in a FASTA format. So they are not side by side as they are in the genome, because you don't know which genome it's coming from. So when you're sequencing it, you're pulling a fragment, you're sequencing it and calling it sequence number 1. Pulling another fragment, sequencing it, calling it 2, calling it 3. So they're all coming one below the other in a FASTQ format. We talked about it yesterday. So when I'm talking about sequenced reads, I'm talking about a FASTQ file that will get from the sequencing. Another source for FASTQ file I do want to tell you is called an SRA database. Let me pick a pen here quickly. Okay, it's called SRA database. I think you all have to look at these databases. SRA, S stands for Sequence Read Archive. This is in NCBI site. National Center for Biotechnology Initiative, Bioinformatics Initiative. So this NCBI everyone must know, because all of you are familiar with BLAST, all of you are familiar with NR database, sequence database. So when you go to NCBI site, there is a place called SRA. Right in the bottom you can see, or you can even type SRA right of the slash. We'll do that in the hands-on series. This SRA database is home for many types of NGS data, from many types of NGS application, and metagenome is definitely one part of it. Okay, so you can get some metagenome application data set. So you will get FASTQ file. You can download it if you want to learn something. Okay, I'm telling you one of the funniest thing that happened in 2010, actually 9 years ago, when I was teaching the first batch of students, and the previous batch had just joined me to do some. We didn't have the sequencing machine at that time, so we were really capitalizing on this SRA database to download data. One of the applications that intrigued us at that time is the windshield wiper in the car. You got the windshield wiper, and windshield wiper kills a lot of small bugs when you're driving fast on the freeway. So what happens is, when you wash off the windshield wiper, that that solution has many insects that we didn't even know they existed. Very small, tiny flying insects. So you could wash it and extract the DNA and sequence those insects that you have never seen, you have never classified. So it was called windshield wiper metagenome. And it was a huge database. And so, one of the students got very excited and downloaded it. We never ever really analyzed it because we didn't have the computers to do it. You really need big computers to analyze this data. So we just downloaded it, and we thought we could analyze it, but we could not. So the reason why I'm saying that is, the application of metagenome is really crazy. It is just your imagination. If you imagine something and you can, you want to know the DNA sequence, like air, for example. Lot of pollen goes in the air, and in Sweden, people were sequencing the rainwater. They were harvesting the rainwater right after spring, and then that rainwater will have pollen from many plants, and they will do metagenome sequencing. So really, metagenome is a concoction of many organisms. The sample has more than one organism, and you're sequencing it, so you can analyze it in the computer. So that's pretty much what it is. So there are two things we can do. Once you get the sequencing reads, you can do two things. One is alignment-based, which is what we pretty much do for every genome. This is common to this portion, really is common to all genome sequencing. It can be human genome sequencing, it can be a single plant nuclear genome sequencing, it can be yeast genome sequencing, whatever it is. So this part is really common. So when we're talking about alignment based, it is a very well developed technology, because we have been sequencing and assembling genomes for a long time. What is new in metagenome is on the right. Why we have to do the binning, why we have to classify them, and how do we compare them and classify, that comes in, that's unique to metagenome. So let's start from the left. Okay, alignment based. When we're talking about alignment based, you in the second semester, you must have done a lot of alignment: BLAST-based alignment, FASTA-based alignment, dynamic programming algorithm-based alignment, multiple sequence alignment, hidden Markov alignment. So you did too many alignments in second semester. All those alignments were different than what we're talking about here. Those alignments were to find homology, similarity between two different proteins, two different DNAs. Okay, so we used the mutation matrix. You must have heard about PAM matrix, blossom matrix. So those matrices told them the distance between two proteins and evolution between two proteins. But here, when we're talking about alignment, it has nothing to do with homology. It is identity that we're looking for. Meaning, this alignment is almost equal to text search, like in Google. Google, when you're text searching, when you make a typo, it will correct it. But it's going to look for exactly the same string. So here, it's not a word or anything, but it's going to look for the same DNA signature. And that is what we're calling as alignment. But you may think that, oh my God, that's really simple. Why are we making so much fuss about it? But at the DNA space, because there are only 4 letters, in English you have at least 26 letters. In proteins, you have at least 20 amino acids. But in DNA, you only have 4 bases. And these are really long string. These really go to millions, right? So the possibility of having the same string coming again and again is more. So, and also this kind of string search happens um, in millions of reads have to be aligned to millions of other reads to be able to find where they overlap. The computational footprint is too huge. So people had to develop algorithm to contain it within the existing computer footprint so that you're not assembling a genome which you took 10 days to sequence and you're assembling it for the next 4 months. Nobody wants to do that because at least it has to have alignment and assembly tools have to be compatible with the time something takes to sequence because by the time they have sequenced another one, you still haven't assembled the old one. So the pace has to be matching. So that's why we have a lot of alignment tools that have matured and developed. So we are going to do that one by one here, and that's called alignment based. So once we have an alignment based assembly, yeah, we now can do, okay, do I have a completed novel genome here? In this particular case, in metagenome case, there are many small microbes sitting there, right? For some of them, you could have a completed genome. We need to identify which ones are complete and which ones are not. Remember, the microbial genomes are circular, right? But your FASTA file after assembly, it will be only linear. So you don't know whether it completes a circle or not. So that's a thing we have to deal with here. And then of course, one could also directly go and from the assembly and annotate it. Find out the 16S RNA, some protein biomarkers, some pathways. Pathways usually in metagenome, people are interested in finding antimicrobial proteins. Or you may be looking for some enzymes from hot spring so that you now have an enzyme that can be stable at higher temperature and do a reaction for you. So other things you can look for in these the first pass assembly is CRISPR spacer. CRISPR spacer, you've heard about it, I've heard about it, everybody has heard about Cas9, right? But in the lab you will do this. You will take an assembly, already assembled file that I have. You can use that to find out CRISPR spacer, how they are put in within the microbial genome. They repeat so beautifully, amazing. We have found some files. We have found 27 CRISPRs repeating with the spacers. You can see the CRISPR is repeating nicely 27 times, and the spacer sequences are different, of course. Okay, so that really explains what you kind of feel for CRISPR-Cas when you do these kind of alignments. It was a very simple blast with the CRISPR and spacer database you have to do. Those things you will do. In protein biomarkers also, you can look for some metabolic pathway. From one soil to another soil, you want to look for some metabolic pathway to know whether this soil is good for this or this soil is good for this, which soil needs some kind of nourishment to be able to grow something. So those are things you can do. So, and then of course, composition based. The next one is composition based. This is unique to metagenome. Okay, composition based. I'll tell you after I finish with the alignment thing. It's pretty interesting. What we're talking about when we're talking about composition, you're talking about the nucleotide composition, not just the GC content or AT content, but you're saying that a given tetranucleotide, how many copies of a given tetranucleotide is there in a given stretch of the genome, the stretch of the sequence. So based on that, you create a vector. I'll teach you all that. I'm just saying in short that is totally different than the alignment based. So you have two ways of handling metagenome, and they nicely converge into giving you a comparative analysis of two metagenome from two different soils or two different conditions. If you are taken it from two gut microbiome, one with disease, one with normal, you can compare those things very well after this. Go to the next slide. Okay, so what's going on now? So what's going to happen is, up to now it was like a linear thing. You learned what metagenome is and you're going to sequence it. Now, what's going to happen is, from raw reads, when I'm calling raw reads, I'm talking about the FASTQ file. You got a FASTQ file. And from there, you're going to get to the assembled genomes. When you do that, there are a lot of concepts we need to know. Coverage, how to assemble a simple alignment-based assembly. What do I mean by alignment? And then bubbles and branches. During alignment, what kind of adjustment we have to make so that we don't get bubbles and branches. We want one linear sequence, right? We don't want it going like this and branching out and not knowing which one is the right assembled genome, right? Assembled genome finally should be linear, right? And then graph theory, how we finally ended up using graph theory, how graph theory applies to assembly, and concept of k-mer was developed. This k-mer, you will hear it everywhere. Whenever you talk about assembly, you will talk about k-mer, no matter what you're assembling. No matter whether you're assembling a genome, metagenome, whatever, you need k-mer. Because without the concept of k-mer, we could not assemble the genome today. Even with the most loaded computer, with even 1 terabyte or 2 terabyte or 5 terabyte of RAM, you cannot assemble the genomes because we get so many fragments. So you need, the concept of k-mer is important. And then finally, there's a method called de Bruijn, which really made sure that with the current existing computers, we can handle the genome assembly problem. Once you finish all these concepts, then you have an assembled genome. So it may look like we're detouring, but you know where they all connect and why we are doing that, right? And then you have, after you assemble the genome, you got this N50, L50, which we use to say, how good is my assembly? That person A has assembled a genome using some other methods, person B has assembled the genome using some other method. Which one is good? How do we measure the goodness of assembled genome? Okay, completeness or goodness. Okay, that's N50 and L50. We'll talk about that. And then we know we got the assembled genome at this point. But in metagenome, we still have to continue after that with the composition-based binning and something like abundance-based correction. I will tell you why abundance is such an important thing, okay? Anyway, so we'll get to here slowly one by one. I'll talk about each one of them, and hopefully we can finish all these within 1 or 2 lectures. If not, we can extend it. That's not a problem. Okay. So alignment-based assembly. So this, again, as I told you, alignment-based assembly is common to assembling either a single organism or metagenome. So if I take a plant and I'm sequencing the nuclear genome, the same methods will apply. Okay. So these are in fact developed for a single organism, which we're also going to be using in metagenome. Yeah. So yesterday we talked about this. Um, I mentioned that when you take the chromosomes from within a tissue, within a cell, you have large number of cells. Each cell will have exact exact copy of the same chromosomes. So I want you to do one thing for me, okay? Uh, I want you to find out if you are getting from human genome. Okay, in human genome we have 3 billion. Write it down. Okay, write it down. In human genome we have 3 billion base pairs. Correct? Yeah. Okay. And then of course they're diploid, so it will be even double. Okay. So when you're getting one microgram, write it down, 1 microgram of DNA from the human sample. Can you tell me how many cells it may be coming from? You got the question? Yeah? You have extracted 1 microgram of DNA from your human tissue, and you know that the total content, how many bases are there within each nucleus, right? Forget the mitochondria and everything right at this point, okay? They're probably minuscule, but the nuclear genome size, and the total amount of DNA you got. Can you compute how many cells it might have come from? Give me an approximate ballpark number, because this is the type of question that Google will ask before they hire you. So you should be able to compute that. Okay, do that as an exercise, and send it to me, or I'll give it to Afia, so I will know who got it right and who didn't. Okay. So now, as I said, from multiple cells you're going to get the DNA. Suppose these blue ones are the forward strand from one each cell. So the top one is coming from cell 1, the second one from cell 2, third from 5 cells here. Okay. Of course, you're going to have hundreds of thousands of cells, but I'm just, for example, drawing only 5. And the bottom, I have the 5 reverse strands. I'm not going to worry about reverse strand at this point, because reverse strand and forward strand gets treated very similarly. It's just a complication right now. For the concept part of it, we don't need to worry about that right now. So, okay. But you're going to get both forward strand and reverse strand, and you're going to fragment it. And the vertical line, orange lines have drawn, each molecule got fragmented at a different place. Second molecule got fragmented here. If every molecule got fragmented at the same place, there will be no overlap between the DNA. Correct? Between the fragments, you will have no overlap, because all of them are cutting exactly at the same place. They won't overlap. So, but fortunately, the way we fragment it, sometimes we just heat it up and fragment it. So, one of the technique for fragmenting, I think, is just heating it up. Okay. So, because of the different DNA molecule break at different points. If you make an enzymatic cut, then all of them will be cutting at exactly same place. You're not going to do the enzymatic cut. You're doing just heating. So they randomly shear here and there, which is good. Now that they got randomly cut, now DNA from the second DNA fragments from here, more likely to overlap with DNA fragment from here. Right? Because they are not sheared in the same place. Okay. So now you got the fragment. You got all these fragments here. Many from forward strand and many from reverse strand. Okay, let's not worry about reverse strand, because reverse strand is nothing but like forward strand, right? If you are given a reverse strand, would you be able to compute the reverse complement? You can always do that, correct? But when you're sequencing it, at the read level, you don't even know where the fragments are coming from. Are they coming from reverse strand, or are they coming from forward strand? You wouldn't know. It will all be sitting in a FASTQ file like this. I have color coded for our own concept only, but you have no idea the first one came from first and third came from forward strand and the second came from reverse strand. You have no idea. This is only conceptual at this point. Okay, so all you have is a large number of sequences sitting there. Okay, so you got a FASTQ file now. You fragmented it, you sequenced every fragment and you got a FASTQ file. Okay, this is what I was talking about yesterday, this kind of fragmentation. And then someone asked whether we fragment it exactly same size. We actually size select. We waste a lot of DNA. So that's why your process, companies, when they're optimizing the procedure, they'll make sure that the fragmentation procedure gives you maximum a number of the size of the sequence you need. Like a 300 base pair, then the fragmentation procedure to maximize the fragments that are 300 size. Otherwise, you'll be wasting the DNA. Correct? Okay. Now, you got reads. Okay, you fragmented the chromosomal DNA from cells, from tissues, and then you want assembled contigs. So this is what you got. You got a FASTQ file. Blues are read from forward and reds are. We don't know which one is blue or red. I'm just saying it just so you know that not all of them are coming from the same strand. Okay. So I'm even going to drop for learning how to assemble it. I'm even going to drop the red ones. I'm going to just assume that we only got the forward. Okay. So I'll first read. I'll start from, because I have nothing. All I got is the FASTQ file. I got nothing, and all I know is how to program it. Or I have a program that can do the assembly. Suppose I'm going to write a program now. Okay, you're all learning programming. So one of the exercises also could be that you can find overlap between two reads by using computer programming. Okay. So let me take the first read. So where will I start? In a computer, suppose I had 10 reads. Okay. And I have a file, FASTQ file of 10 reads. I'm going to start writing a code which will say that take the first read. I'll do it in a loop, for loop or whatever programming language you use. There will be a loop. You will say take the first sequence and find where they match with the rest of the sequences. So when you have 10 reads, you're doing 10 searches, right? First sequence with everything else, second sequence with everything else, third sequence with everything else. When you're doing that, when you have 30 million reads, then you're doing 30 million times 30 million searches. That is where the computer become too overwhelmed. It gets very slow. Why I was telling you about the methodology that has been developed to take care of it. KML, everything. So, but right now we're going to go simple alignment. So the first read, I just make it little enlarged now. So I'm looking at, this is the green sequence. I just color coded a stretch of sequence green and I color coded the stretch of the sequence red. Now I'm going to look in the rest of the blue reads, because I just dropped the red reads for convenience right now. In every read I found out the 5' end and the 3' end. So that's what you're doing. We're doing the 5' end and the 3' end. Is there similarity in the 5' end? Is there similarity in the 3' end? If there is similarity in the 3' end, this is 3'. No, I'm not writing it well. 3'. Okay, and this is 5'. Okay, 3' and 5. So to extend one means that this is one read. To extend one, I need to see whether something overlaps at the 3' or 5'. So here I've put a lot of constraint here. These are all the constraint you will have in your tool, in your program that will write. Duplicate read, don't add value, correct? If there are two duplicate exactly same, then it's not going to extend anything. So I can remove it. That's the first thing I'll do. Non-overlapping read, don't add value. So for example, if I'm taking one, if none of them are overlapping, then it's adding no value. Okay. Match in the middle of the read, do not add value. Meaning that say one has a red in the end, and see the sequence number 3 has a red in the middle. that kind of match is not of any use because you can't extend it. You're going to have to see where this blue will go because this blue is not the same as this blue. So it can't sit there. This is different. So this won't work. Okay. So and then match 5' to 5' do not work. So if you have a green here and you have a green here, that's not going to work because it has to have the directionality. 5', 3' will match the 5' of the other and extend it. Right? That's how it goes. So if the sequence is like this, 5', you know the 5', 3', right? So the sugar phosphate chain is unidirectional. You can't come back like that. So if the 5' and 5' matches, so this 5' and 5' is matching here, let's say, matching. But after that, it's not matching. Then you can't even extend it. It's just wrong. Okay. So whereas if the 5' and 3' is matching, then you can extend it. Okay. So let me get my pen again. Okay. So tell me now, you can write on the chat, okay, quickly. This is read number 1. You see read 2, 3, 4, 5, 6. You can read that, right? Yeah? I don't know why this became so heavy, this black box. But you see 1, 2, 3, 4, 5 is not there because that was a 6, 7, 8, 9 and 10 are not there. Okay. So which are the 2 reads that can extend read 1? You got to put the numbers right there in the chat box right now. Okay, 5 or 6 of them have answered it. Okay, so it is going to be 7 and 8. I think some are not getting it right, but most of you are getting it right. Why 6? Some people are putting 6 and 8. 6 has a yellow 5', and we don't have yellow in read 1. Okay, so 6 cannot be the correct answer unless you are not seeing the number right. It is 7 and 8. Okay, so how is it going to extend now? 7. I'm going to draw it here. 7 will go here like this, and it will have a red one here. So red one will collapse now into one common area. Yeah, and then 8. This is 7, and 8 will go here. Sorry, it's not coming out straight, but that's okay. You get an idea. Okay, and this will collapse. Now you got a long reed from here to all the way here. This will be a contig now. You put together a long contig. Once you do that, now you take this contig and search everything else. Then you go iteratively like that until this contig can be made bigger and bigger and bigger. Okay? Once it's no longer able to be bigger, for example, let's see. 7 has no end at all. Okay, I didn't put that. So okay, that's okay. So you're getting a hang of it, right? Okay. So that's how you do that's how you do read by read assembly. That's what we did for Sanger sequencing. These are the method we did for Sanger sequencing. Nothing more fancier than what we're doing here. Okay. So now here I'm just showing much more complicated why you don't get a complete chromosome. Okay, in this case I'll tell you why you're not getting complete chromosome. So I have put this colored end here. So now you know that okay, if I take this reed, I put it here, and I was able to extend it using this one on the left. Okay, and then I used this to extend on the right. Okay, then I used this one to extend further, right? And then I got an yellow end. If I got yellow, I could do here. Okay, but I did not find anything from this end that matches any other reed. And those are dead ends, you cannot extend it. Unless you find both ends, 5' and 3', something that's matching, otherwise you end up in dead end. You end up dead end here. So this will be contig 1. You can do anything because either you didn't sequence deep enough, so you didn't get fragments properly, or just the sequencing technology itself has a bias and did not sequence further. Okay, so you got contig 1, but that's okay. But still, you have other things left. Now you can start with this one. Okay, and then you can extend with this one, and on this side you can extend with this one, right? So now you got another contig, and now you have a dead end here and a dead end here because nothing is matching. Now you got contig 2. So what this exercise is telling you is that in spite of getting a lot of reeds, many reeds will not have the end, both ends like a dumbbell. It should have a signature on both ends matching with something. If it didn't, then it won't extend. It will stop right there. Okay, so when that happens, you get one long assembly, but it's not continuing. So even though the DNA came from one microbe, you know that one microbe, the single microbe, you got all the reads from a single microbe in a microbial genome project, not metagenome. You took a microbe and sequenced it. Still, because the sequencing technology gives you unbiased, gives you biased reads, you still may get 3 or 4 contigs. You should ideally get only one because there is only one chromosomal DNA in a microbial genome sequencing project. But you still may end up getting number of contigs which are disconnected because of various reasons. I will tell you why, one of the reasons why it might happen. Okay, in the bottom here, just so you missed out on what are these orange and red and I'm putting here. Are they really colored? DNAs are not color coded, okay? They are just sequences. So I'm just saying, for example, the purple one, the purple color, whenever I'm doing, it's the ATGC stretch. Okay, everybody is getting it? What is color code? These are all sequences. Okay, for example, let me erase all this and clean the board a little bit. Okay, so yeah, so this is the sequence that's here. This one. This is this guy. So the red and purple, I just put a conceptual color there. What exactly I mean is ATGC. It was there in ATGC in the 5' end. Then I color coded them as purple. Whenever it was TGAC, color coded them red. Whenever it was GCTG, color coded them green. Whenever it was CGTA, color coded them yellow. When I did that, that's what you got. This is what we got. Okay, so when you're collapsing, when I said that now collapse it, right? This becomes collapse it because they're overlapping. So you don't need 2 parts of it. You're collapsing, you keeping it from only one sequence. Other sequence, you're merging it. Okay, so when you're collapsing it, this is what happens. So you only keep one ATGC. So this goes black, ATGC, and you shift to this. And then TGAC, and then you shift here. You shift from here to here. You miss this. You shift from here to here. You miss this. So that this is your contig now. That's exactly contig 1. You got it? So when you're writing program, this is what you need to do. Overlapping region, you take it from only one read. Okay? And then stitch it and you get a contig 1. I'm hoping everybody is getting it. I'm going into this much detail because even if you are little dozing off in the morning because you didn't get a strong morning coffee, you should be able to get it. That is why I'm emphasizing on these things, okay? Otherwise, these are probably quite easily understandable, okay? But I'm emphasizing it because I want you to get it. When you write a program, you need to exactly implement it properly. It's not enough that you understood it. You got to implement it properly. You now get a contig 1. Similarly, you do contig 2. Okay? Okay. So now I'm going to go into coverage. So the reason why I'm switching into coverage is that coverage is an important aspect of why sometimes you do not get all the reads assembled into one single chromosome or one single circular chromosomes. Okay, it is still split into contig 1, contig 2, contig 3, and you still don't know contig 1 is left of contig 2 or contig 2 is left of contig 1. You don't know that because you don't have a reference genome yet. Okay, so coverage is a very important concept in entire genomics. People will talk about coverage all the time. What coverage did you sequence? What do they really mean by that? So let's go through the concept of coverage right now. Okay. To understand coverage, okay, yeah, someone, before going to coverage, I was checking whether someone has any question, and I do see a question from Khan. Do we need to remove duplicates? Okay, duplicates removal is good because then you're saving time. I will tell you how many duplicate reads come after Illumina sequencing. Sometimes 20 to 30% of the reads are duplicate. So if you have 30 million reads and 20% of them are duplicates, of course you want to remove them. Because why would you want to keep searching again and again? The loop can go into 20 million loop is much faster than 30 million loops. You always remove the duplicates just because of the computational footprint. You want to keep it safe because these are large numbers we're talking about. Okay, they don't hurt the assembly, but they hurt the computing time. Okay, I'm going to come to overlap. That's where the Kmer comes. Okay, how many bases you overlap? One of them is asking how many bases you use for extension. It cannot be 2 bases, you know that. It cannot even be 4 bases as I showed in the example. Okay, why? Because 4 bases, there are only 256 unique 4 bases, 4 to the power 4. 4 base types and 4 to the 4 ways they can be different, unique. You have 256 unique formers and then you have 30 million reads. So imagine by accident how many millions of reads will have the same former on a 3' end and the same former on the 5' end just by accident, coincident, right? So no, former is not a good answer. Although I used that in the example, I'll tell you in the camera. Usually it is 31 mer, 31 through 60 mers. But there is a penalty you have to pay. If you increase the overlap size, extension, overlap size, if you increase it, you can't increase it too much because then you're going to lose a lot of reads because you will never find overlap. Okay, so Kmer is an important concept. I told you I will be talking about it, so I'll come back to that. So there is a third question here. Isn't it possible to get 2 fragments with same 5', 3'? Yes, that's what I talked about. When you have very small overlap size, then yeah, you're going to get bubbles and branches. We're going to talk about that as well. One of the concepts, remember, we had bubbles and branches. So the overlap size has to be very smartly picked. Okay. In order to determine the duplicates, we still need to make a large number of comparisons. Wouldn't that affect the computation time? Yes, correct. It will affect this computation time. But, think in your mind, in your brain, If I give you two overlapping sequences, two duplicate sequence of 100 bases each, I used to give this as an exercise in the class when I used to do sequence alignment, but I don't have that now. But think of it, how would you, I give you one sentence and in the next page there's another sentence, many sentences, but one of the sentences is exactly what is there in the first page. Would you compare word by word the other sentence? Is that your brain will work very smartly, right? It will compare the first word with the first word, and the last word with the last word, and the middle word, and actually size of the word. All that will compare and somehow quickly figure out. So if the first word, so for example, suppose you have a lot of sentences in the second page, one of the sentences is exactly what you're looking for. Okay, I'm going to look for the first word. How many sentences in the next page has the first word this? Many will fall apart, right? Only three of them has the first word the same. Then I'm asking, okay, the last word. Then there's one sentence. Boom, you can take it out. So you can reduce it. You are not going to compare letter by letter. You're going to be smart about it. That's the implementation details. Okay, so there is a tool called Picard tool which does the duplicate. They do it very efficiently. It's all about computational efficiency. Yes, that is that's a problem here. Okay, so okay, so I don't have any other questions, so I'm going to move on. So keep the questions coming. I'll take whenever the break comes. I'll answer it. Okay, so you got the sequencing. So one of the ways to understand what coverage means is easier to understand it if you think of a reference genome. So to understand the concept, I'm going to use a reference genome right now. I'm going to assume that the model or the organism that I just sequenced has a reference genome. Okay, so now I'm going to map all the reads that I sequenced. There's a tool called bowtie. We will talk about it later, bowtie and algorithm and everything. But bowtie is a tool that maps, that finds a home for a fragment, complete fragment matching the reference genome. It matches the full fragment to the reference genome. It says that, okay, this read, the 15th read came from the beginning of the genome, and the third read came from somewhere here in the genome, and the second read in my FASTQ file is coming actually from somewhere in the middle. Okay, so it's finding home for everything. Okay, this is how the reads are mapped now. So this is all the number of reads I got. This is a very simplified thing I'm showing just for concept's sake. Okay, now I'm going to the next slide. Okay, so coverage is, you have the chromosome on the top and you have mapped all the reads, correct? These are the chromosome, these are the reference genome you have, and you have mapped all the reads. Okay, so at every position in the reference genome, suppose the reference genome is of size 6000. Okay, so at position number 1, how many reads are passing? At position number 2, how many reads are passing? In the human genome, you will have a 3 billion position to interrogate. Okay, 3 billion positions you have to have reads crossing. Then only you can say that I have sequenced the whole genome. Okay, so here in the hypothetical case, there is a genome of just 6,000 bases. I'm going from left to right, and at every position I'm asking how many reads are crossing that. So I went to this position. Okay, and I'm asking, I'm drawing a vertical line, and I'm saying how many reads are from this region got sequenced. So how many reads are intersecting this? There's 1, 2, and 3. So this will be 3. So I'm moving from left to right and that's asking at every position. I'm not going to go every position. So the other position I'm taking here, how many reads are crossing? 1, 2, 3, 4. So 4. So this is called coverage. This is the coverage of sequencing you have done at this particular position. You got a coverage 3 here, you got a coverage 4 here. Of course, coverage varies all the time. It shouldn't, ideally it should not vary, but If you don't sequence deep enough, it can vary considerably. Okay, so here we haven't sequenced deep at all. So this here, there is only one reed crossing this. And then you have regions that are not covered at all. Okay, either because you haven't sequenced deep enough, or maybe this region never gets amplified in the sequencing when they amplify the cluster formation, when they're forming clusters in the sequencing machine. Maybe this region has a sequence that it doesn't like, it doesn't amplify it. So, this is called coverage at this site. So, if you do this for every position, at the 6000 position, if you calculate all this and sum it and divide it by the number of positions 6000, you add all these numbers divided by 6000, that will be your coverage. Okay, for example, here right now we did only 6 position. So you can say that 3 plus 4 plus 1 plus 0 plus 5 plus 2 divided by 6 will be the average coverage of this chromosome. Okay. So this is very circular because you had to know the reference genome to get the coverage, right? But how, you're not going to map. You don't even have the reads yet. How are you going to compute the coverage? The sequencing lab will ask you how much coverage do you need? How many bases you want me to sequence? So you don't have the reads to map and tell. You are asking for the reads, right? So there must be a better way of doing this. But you understand the concept of coverage, right? Okay. In the next slide, again the same coverage. So I think this is redundant. I must have done it for some other reason. Okay. So now you have the coverage without a reference. How would you do that? Think about it now. Okay. So you know the read length. Okay. You're randomly picking. Okay. Again, I'm going to have to ask a question, and I think I'm going to 9:30. Oh, it's only 10 o'clock. I have 15 more minutes. Okay. So read length. You know the read length. They're saying that you're going to order, saying that make all the reads 100 bases long. So you know it. That's your input to that and those people. And okay, so you got a FASTQ file with 100 bases, and the number of reads are 10 million reads are there in that FASTQ file when you got it. Let's say, okay, you got 10 million reads, and they gave you 10 million reads of 100 bases. Okay, how many total number of bases that got sequenced? Are you with me? Total number of bases will be 10 million times 100, correct? That's the total number of bases sequenced. You need to know how many bases totally got sequenced. Okay. And then I'm saying, you know the organism you gave for sequencing. Even if the reference genome is not there, the size of a genome can be estimated in the lab using FACS analysis and other molecular biology tools. You can estimate the size of the genome. Sometimes in the published literature, they will give you the size of the genome. So you know your genome is 10 million bases long. Okay, and you got 100 bases read, 10 million 10 million reads. So the coverage is, what is the coverage of the coverage that is sequenced here? Can anyone answer quickly? Okay, 100x is the right answer because the reads are sitting randomly. So you would say that if you're aligning 10 million reads, 10 million reads to a genome of 10 million length, each position will get covered 100 times because you have, it's 100 bases long each read. So it is 100x coverage. The answer is 100x. This will repeat many times. If you didn't get it, don't worry about it. Okay. So, for example, now I'm telling that my genome is, say, my genome size is, I'm going to give you another problem. Okay, I have two questions here. I will answer them. Okay. Um, um, Rohit's question I'm not getting, but I'll come back to that. I'm going to give you one more quick tool. Most of you have answered 100, but this is only 10 people have answered. But I'm going to give you another one. Okay. Suppose human genome. You know that human genome on 3 billion bases. I want 30x coverage. How many bases will I sequence? Question number one. Question number two is, if the read length is 100 bases, read length is 100 bases, then how many reads? Can you answer them quickly? First answer, how many bases, total bases you will order? Because that's the first question a sequencing guy will ask you. How many bases you want, ma'am? One more. 90 billion, yeah. 90 billion is the right answer, Rohit. 90 billion is the right answer. Okay. So if the read lengths are 100, how many reads will you expect in your FASTQ file? So when you answer 2 comma 1 comma, so because I asked you two questions. The first one, many are getting it right. I see the same set of people answering. Okay, that's not good because I want everyone to answer. Make an effort. If you don't get it, can you ask me a question? I'm not getting it, ma'am. You just have to say that I'm not getting it, ma'am. So I will try to say it in a different way. I will give you some other example. Okay. So question number two: how many reads will be there in your FASTQ file if the read lengths are 100? Nine. Yeah, 900 million is the right answer. Okay, you will have 900 million reads in your FASTQ file. Do you see that? What I'm talking about? These are really huge files. So if you really ordered human genome sequencing 30x coverage, 100 bases per read, your FASTQ file will have 900 million reads. You know the FASTQ files has 4 rows per read, right? I showed you yesterday. Yeah? So how many 900 times 4? That many rows it will have. These are really, really huge files, so you get a feel for it. And 30x is not a lot. 30x you need for germline mutation. You need 100x for assembly. So you can imagine how big these files will be. Just to get an idea, I'm talking about compute power, I'm talking about computational footprint. You can imagine what kind of computers and what kind of optimized algorithm you would need to fit these things in a computer. Okay, that gives you some feel for this. Okay, so people who are not getting it, please don't be shy because nobody's going to evaluate you, but you need to follow it right now because these classes will get more and more harder. These are basic concepts you need to know so that you can understand the next step. Okay, so don't be shy. If you get the answer a little late, just type it in. Okay, it will keep coming here. You're not holding the class for that. Okay, so I'm going to assume, because nobody said I didn't get it, I'm going to assume that everybody understanding what a coverage is and how will you compute a coverage when you're given a FASTQ file and you know the genome size, and how would you tell a sequencing person how many bases do you need, what do they need to sequence, how many lanes they have to put your sample so that you get um, enough bases sequenced for your sample. Okay, so next, so going back to that branch, I said all these concepts, bubbles and branches. This was one of the questions that I think was asking. Chaudhary was asking. Okay, she was asking a different question. This was the question people are asking. Okay, 4 bases. Wouldn't there be too many overlaps? And one of the questions I hear is coming. That's a little tougher question, but answer will come later, because many microbial species in metagenome will be very similar, right? They're very closely related species. And that is the reason why I said just the sequencing based, alignment based assembly is not good enough for metagenome. Okay, and there are very many methods people use to correct for closeness of the species, why you will form chimeric thing, because this species kept assembling and it picked up from the closely related species and went back into that species. So it will be chimeric reads. How do you break for the chimeric reads? Those are stuff we'll learn a little later. But now, right now, we are just talking about alignment based assembly, which applies to any genome sequencing, not just metagenome. So we'll wait to answer those questions later. Okay. Then you have bubbles and branches. Okay, so overlap of 4 bases. I did give this as an answer to one of the questions. If you just have 4 bases, what will happen? Total unique overlap will be only 256, right? But the total number of reads are this many. You're getting from it's a big genome, and you got 10 million reads, let's say. So just by accident, okay, 10 million divided by 256, that is how many 10 million divided by 256. That is how many reads will have a 3' and a 5' same. So you're just going to be extending it in all possible wrong directions. Okay, so it won't work. It was easy to show you 4 bases just to get the concept right. Okay, so you need an optimal overlap. Okay, here I'm just showing it as an example, 4 base overlap, right? For example, let's say you had this is the red one. Again, I'm putting color code for the former. Okay, each color is a former, unique former. Okay, so now you have green and red dumbbell, then you have a red and blue dumbbell, then blue dumbbell and open one, and then you have a red here on the 5' and a purple on the 3'. Then you have a red here and the green on the 5'. So when you are assembling, when you're using this read, to find out which ones are overlapping, the 3' is overlapping here, 3' is overlapping here, 3' is overlapping here. So when you're extending it using your tool that you wrote, remember we conceptually wrote a tool, how in the for loop you can take the first one and keep asking, okay, this is the 3', how many has the same and the 5', then I'm going to extend it. What's going to happen? Then you're going to have 3 of them. You don't know which one to take. You're going to make one contig with the red blue dumbbell. You're going to make another contig with the red purple and another contig with red green. Okay. So, and then you don't know how it's extending. Then you go, there is another dumbbell with a green and blue end. Okay, so the green and blue, the end of this is the blue. So this is green, so it's going to make a bubble. That's how the bubbles are formed. And then you will start from this blue and keep extending it one way. So there are bubbles formed, and then you have too many branches. Genomes are never branched, right? They're one linear sequence. So when we are assembling it, you have to get one as much as possible. You don't want branching happening. You don't want bubbles. You want to clean for that. So 4-mer will not work. We know that. 5-mer will not work. 6-mer will not work. How do we decide what mer will work? Right? One of the simple thing I'll tell you, take a 10-mer sequence and blast it against NCBA blast. Okay, it won't even give you any result because it's going to say that, the score will be so small that it won't give you any result. So you keep extending your query nucleotide sequence, and somewhere close to 25 to 30 or 35, you will start finding hits to the database. And at one point, you will start finding unique hits. And somewhere in 31-mer, it starts finding unique hits. It's unique to a given genome. Okay, so that, like, more or less you got it empirically. Okay, but that's okay. So by trial and error, by empirically doing this, people know that somewhere close to 31-mer, and of course, longer is better. But what is the shortest that can be unique? Comes to 30, 31, 32, 33. That's where it comes to. So that's a critical overlap that we're going to use. That is also called a K-mer. Okay, so I think I have only few more minutes to go. We got the coverage concept right, and many of them kept typing 9 million, 9 billion, 90, 10 to the power 8, 9, 10 to the power 7. So coverage, we got the concept of coverage, we got the concept of bubbles and branches, and the questions were good because yes, in metagenome we have very closely related species, and that's a complication of metagenome which will come at the composition based assembly. Right now, we were just talking about alignment based assembly of a single genome. Okay, so which is also used in metagenome, and the cleanup happens afterwards. Okay, so we did that, we got some concept ready. Now we are going to use in the implementation. When they implement these things, it's complicated. You saw that 900 million reads you're going to have if you sequence human genome or any other mammal that hasn't been sequenced yet. Most of the mammals have 3 billion bases. So if you sequence them, you have a file that's 900 million reads. So the implementation has to be done good. You got to weed out many things that you don't need. Removing duplicate is one thing. How, without spending a lot of time, how do you remove duplicates? How do you remove other connections, other non-overlapping reads? How do you reduce the graph so that the computer footprint is smaller and smaller? That's what we will learn in the next lecture, which I think is next Monday. For now, I think we are done. Unless you have any questions, I don't see any more questions coming. So, I don't want you to be late for the next class because I know you're sitting in the lab and you have to go to your classes. Why don't you take a break and go to your classes, and we will continue with graph theory next Monday. Okay, bye now.